Welcome to Lab 4. In this lab, we are going to be discussing the forces of evolution. So the first thing we're going to talk about is what is evolution overall? So evolution, the general definition, the easy definition, is just a change in allele frequency. So straightforward, really easy, but really complicated at the same time. So there are some important implications that we have to think about when we are talking about evolution. So first, we have to keep in mind that this is discussing the genetic level, not just the phenotypic level. So because of the complex relationships that often exist between dominant and recessive alleles, evolution may be occurring subtly at the genetic level without us actually realizing it at the phenotypic level. Right? So it might be happening, but we won't physically see it because, again, it's happening at the genetic level. This is particularly the case with polygenic traits, where we have multiple genetic loci at play at any given time. Right? Secondly, evolution involves allele frequencies. So these are ratios of different alleles for each trait and are therefore then calculated for groups rather than individuals. Therefore, evolution happens at the population level, not individual level. Third, because evolution is about allele frequencies, evolution occurs in traits. So within a population at any given time, some traits may be evolving while others are remaining the same. It's not that the population isn't evolving. Instead, it is that the trait or traits are evolving within a population. Finally, evolution is a change in allele frequency. This means that evolution occurs across multiple generations. To measure change in allele frequency, we must look at populations over time. If a generation has an allele frequency of X for a trait, and you see that either the parental generation or the following generation has a different frequency, then you can say that the frequency is changing and evolution is occurring or has occurred. There are many things that drive evolution, and most often they'll act simultaneously in real life. So again, there's many factors that can cause evolution. The first one is genetic recombination, right? We learned about that in the second lab. Genetic recombination, again, means that we are exchanging genetic information during that crossing over phase of meiosis. Therefore, what was originally given on one chromosome may not be the same after this phase and will have resulted in a shift in genetic information. Again, remember, no new genetic material is being created, but new combinations of existing genetic material are produced. Recombination is not considered one of the traditional forces of evolution, but it does play an important role in creating variation, which is the raw material then for the evolutionary process. So aside from genetic recombination, we want to look at mutation, natural selection, genetic drift, and gene flow. And I'm going to go through each of those with you. Just a side note, for each one of these, and specifically, well, really for natural selection, genetic drift, I have separate videos that are linked in this PowerPoint. You go ahead and when you look at the PowerPoint, click on the link. They're very, very good videos done by the YouTube channel Amoeba Sisters. They're animated, they're very entertaining, and I highly suggest watching them because if you are a visual learner like I am, then seeing those videos can be really helpful in actually understanding what I'm talking about when you might find it a little difficult by me just lecturing at you over a video. So that said, let's start with mutation. While recombination, as we know, results in new combinations of genetic material, it does not mean it creates new genes. The only way you can have new genes created is through mutation. So mutation, the generic definition, is a change in the genetic code, right? Any change in the genetic code is a mutation. Most mutations occur during DNA replication. Usually, DNA is replicated perfectly, but sometimes there are errors in the replication process, which then results in mutations. If these mutations happen in DNA segments that do not code for traits or play a role in regulating genes, they can have little to no effect, right? It might not even be noticeable. However, if they occur in coding DNA, they can have a profound impact. Mutations can occur in cells all over the body, but the ones that impact evolution occur only in the gametes, right? So in order for mutation to impact evolution or have an effect on evolution, they must happen in the gametes. They cannot happen, or they can. I mean, mutations can happen in your somatic cells, your body cells, right? But they won't impact evolution because your somatic cells, your body cells, aren't being transferred to the next generation. However, gametes are. So if a mutation happens in the gametes, that is the genetic material that is then sent to the next generation, to your offspring. They then carry that, goes on to the next generation, and there you have evolution happening. Mutations can be very small. If a single base pair of DNA is altered, it changes the corresponding codon. This can result in a different amino acid, which can then result in a different protein. These kinds of mutations are called point mutations. Most mutations occur during DNA replication. Point mutations, again, they occur at a single point in the DNA. So let me give you an example. And the example is sickle cell anemia. So sickle cell anemia is a one base pair substitution, and it's substituted by another. And the sickle cell allele is formed. Expression of this mutated gene produces misshapen red blood cells that then cause blockages in the blood vessels. Mutations can also be much larger errors. During the process of meiosis, a large section of a chromosome can be moved elsewhere on the chromosome, or it can be moved to a completely different chromosome. So this is no longer a point mutation. Sometimes a whole chromosome can be duplicated, and this results in an extra chromosome. So an example of this is Down syndrome, where you have an extra chromosome 21. It's also known as trisomy 21, so 3 chromosome 21s. Other times, a whole chromosome can be lost, which then leaves the organism with one fewer chromosome. So an example of this would be Turner syndrome. In Turner syndrome, you have a single X chromosome and no other sex chromosome. Sometimes mutations are caused by external factors like radiation or chemicals. Most mutations, however, tend to be spontaneous with no known causes. Mutations are not always harmful. Many are neutral or even advantageous. However, mutations create new genetic material such as new alleles. These variations can then be acted on by their evolutionary processes, and therefore they work together with other forces to bring about evolutionary changes. So that's mutations. Next is natural selection as a force of evolution. So in 1859, Charles Darwin published On the Origin of Species, and this presented his ideas about evolution, and it outlined his theory of natural selection. This theory offers an explanation for how evolution occurs. It states that beneficial alleles increase in frequency over time in a population because of increased survival and reproduction of individuals with those alleles. The mechanism behind natural selection is, first, there's always variation in a trait and the alleles that code for them within a population. And second, not everyone in the population will have the same version, right? So firstly, again, there's always variation in a trait and the alleles that code for them. Not everyone has that same version. So secondly, in the mechanism, there is a tendency towards over-reproduction compared to the limited availability of resources. In general, more offspring are produced than the environment can support. This then causes competition for resources which are always limited. They may be distributed unevenly in an environment or limited seasonally, or there may be some resources that are better, so more nutritious, safer to eat than others. So at any given time, members of a population are competing for access to these resources. So that's the second. Remember, the first mechanism is there's always variation. Not everyone has these same versions. Secondly, there is a tendency towards overproduction compared to the limited availability of resources. Third, because of this competition, some versions of a trait will be better suited to the environmental context than other versions, like accessing food or reproducing efficiently. Other versions will be disadvantageous, like limiting access to food or hindering reproduction. Finally, the organisms with the more helpful traits will out-reproduce the other organisms and pass their helpful traits on to the next generation. Over many generations, the population as a whole will have more of the helpful traits because they're being passed on at a greater rate, and the population will become well adapted or fitted to the environmental context. So let me recap that one more time, right? Natural selection overall states that beneficial alleles increase in frequency over time in a population because of increased survival and reproduction of individuals with those alleles. And there is a mechanism behind natural selection. First, there's always variation in a trait and the alleles that code for them within a population, and not everybody in the population will have the same version. Second, there's a tendency towards over-reproduction compared to the availability of resources, which causes a competition for those resources. Third, because of that competition, some versions of a trait will be better suited to the environmental context than other versions. This then causes advantageous traits, right? So they might be able to access food better, or they might be able to reproduce better, right? So these are advantageous traits. Which then finally the organisms with those most helpful traits will out-reproduce the other organisms, and they will pass those helpful traits on to the next generation. This is how natural selection works. So important to know is natural selection emphasizes traits and alleles, not the individuals carrying those traits and alleles. So what is advantageous? Those advantageous traits are going to be successfully reproduced and then passed on to the next generations. All right, so that's natural selection. Then we've got genetic drift. And so sometimes allele frequencies will change randomly. And this random change is known as genetic drift. So the definition is genetic drift is changes in allele frequencies that occur randomly due to factors such as differential reproduction and sampling errors. So in order to understand how genetic drift works, it's helpful to think of it as a statistics problem. And again, I know statistics, math, kind of scary. I know I scared you a little bit in chapter 3 with the mathematics. Don't worry. Again, don't worry too much here. I will explain it like a statistics problem. If it doesn't work for you, again, don't stress out. The video is fantastic for explaining how genetic drift works, and I'll give more examples. So, statistically, imagine a population where an equal number of dominant and recessive alleles are represented. You might think that if the members of this population mate, there will be an equal number of dominant and recessive alleles in the second generation. However, when you think of flipping a coin, you realize that this is not necessarily the case. When you flip a coin, there is a 50% chance each time you flip that you will get a heads facing up. The results of one coin flip are independent of the results of the next coin flip. So the first coin flip, 50% chance. Second coin flip, 50% chance. This every time is a 50% chance. So these chances, right, is independent of the next coin flip. So if a coin is flipped 10 times, the results could be 10 heads or 10 tails in a row. The outcome is unlikely, but it is statistically possible. Similar patterns emerge in mating. If two heterozygous individuals mate, there is a 50% chance that the mother will pass on her dominant allele and a 50% chance that she will pass on her recessive allele. The same is true for the father, right? Heterozygous male, 50% chance dominant allele, 50% chance recessive allele. However, if these individuals produce 4 offspring, it is entirely possible that the mother will pass on her recessive allele all 4 times, and the father will pass on his recessive allele all 4 times. This outcome, again, is unlikely, but it is still possible. So we could start with the population of 2 heterozygous individuals and end up with the second generation of all homozygous recessive individuals. This process is called genetic drift because over multiple generations, it may cause a population's allele frequency to drift away from its original allele frequency. At the same time, not every individual will have the same number of offspring. And this differential reproduction, too, can cause random shift in allele frequencies. So if individual A has 5 offspring, and individual B has 0, then individual A's genetic material will then be overrepresented in the next generation, and individual B's genetic material will be underrepresented. Therefore, you now have a slight shift in the population's allele frequencies with more of individual A's alleles going forward. So, I mean, it's like a statistics problem, but I know it might seem really confusing. So again, watch the video. It's very, very, very clear when you watch it. With genetic drift, I just think of it as genes drifting, essentially. Like the alleles frequencies moving around, right? So with genetic drift, there's a special kind of genetic drift, and it's called the founder effect. The founder effect occurs when only a small number of individuals from a larger population are involved in producing the next generation. So this can happen for many reasons, right? A major die-off happens, or a group of people leave the original group and start to live somewhere else, right? So say we're taking the group of people leaving and settling somewhere else and reproducing there. So these individuals will then reproduce in isolation from the original population's gene pool. In cases of the founder effect, the founders, or the individuals involved in reproduction separate from the original larger population, do not necessarily represent the larger population's allele frequency. So when these founders reproduce, they don't generate the original larger population's allele frequency, and in effect, they jumpstart genetic drift, right? So let's see here. This is kind of an example. You have your original population. It's kind of half brunettes, half blondes, right? The blondes leave the original population. They become a founding group. They move to an entire separate region where they're isolated from the original population, and then the future generations, so they're involved in reproduction, the future generations will all have the alleles present in that founding group, and here we see that they're all blonde and there's no brunettes because they're not represented in this founding group. So that's genetic drift. Essentially, the blonde allele drifted and they reproduced, and now you have future generations being only blonde, right? Let's see, the founder effect. So the founder effect can lead to what is called a genetic bottleneck. And so this is when a small founding population does not have the original population's gene frequency, which can then lead to what we call the genetic bottleneck. And so some of your original genetic diversity is entirely lost. So in this case, that brunette genetic diversity was completely lost in the next generation. So I'm going to give you some examples of genetic bottlenecks that have occurred in the history of Earth. We'll give you 4, and then I'm going to go into detail with the 5th one. So the first one is the Wessant. It's also known as the European bison. They faced extinction in the early 20th century. Of the 3,600 individuals alive in 2000, all are descended from 12 specific individuals, and only 2 distinct Y chromosomes are left in the species. So essentially, all of them died off. There was only 2 males left and 10 females. Those 2 males and 10 females repopulated, right? And now, as of the year 2000, we had 3,600 European bison, right? So all 3,600 can trace back their genetic material back to those 2 males and 10 females. The second one is the northern elephant seal. Their population fell to about 30 in the 1890s, although now its numbers are in the tens of thousands. So the northern elephant seals, the tens of thousands that we see in pictures, if you go to see them, every single one of those are descended from just 30 elephant seals. That's all that existed. They repopulated, did a really good job at it in the 1890s onwards, and now we have tens of thousands of elephant seals as opposed to the 30 that we had. So they're a success, right? They were really good at repopulating. And the third one is the golden hamster. The vast majority are descended from a single litter that was found in the Syrian desert around 1930. I don't have the numbers on that one. Just know that they went through genetic bottleneck and they were able to repopulate it. But I don't have the actual numbers on that one. So I know I said 4 examples, there's just 3 examples. The fourth one I will go into more detail, just because, I mean, you might be familiar with elephant seals. I don't know how familiar you are with European bison or the golden hamster. You can look them up. But there is another animal that we are very, very, very familiar with, and you might be very surprised to find out that they went through a genetic bottleneck and almost went extinct. And that is the cheetah, right? So as a species, cheetahs have famously low levels of genetic variation. Like there's so little genetic variation among cheetahs. So this can be attributed to a population bottleneck they experienced about 10,000 years ago. They just barely avoided extinction, right? At the end of the last ice age. However, the situation has worsened in modern times due to habitat encroaching and poaching. So they were able to repopulate, but again, we've got, poaching and encroaching on their habitat, and their populations are declining. So this, again, the declining populations, again, is decreasing the genetic variation of a population or a species that already has very little genetic variation. So this leaves them very vulnerable to extinction. So again, a genetic bottleneck occurs when a population is greatly reduced in size. It limits the genetic diversity of the species because only a small part of the original population survives. With a greatly reduced gene pool, the remaining members of the species may have trouble adapting to new environmental conditions such as climate change, and a genetic bottleneck can be caused by a number of factors, including natural disasters, overhunting, or habitat destruction. So there's two bottlenecks, right? So scientists believe that cheetahs have already survived at least 2 genetic bottlenecks. The first occurred about 100,000 years ago, right? So I was talking about this second one here, but the first one was about 100,000 years ago when cheetahs left America to spread out across multiple continents. This left only small pockets of cheetah populations which were too far apart from each other to interbreed, right? So they left the Americas and they spread out, and because they were so spread out, they couldn't really go back and forth and exchange genetic information. The second event, which is the one I was talking about, it happened about 11,000 years ago here at the end of the last ice age when many of Earth's large mammals became extinct. At this time, the cheetah populations in both North America and Europe died out, and scientists believe as few as 7 cheetahs survived in Africa. So if we thought that the just 12 individuals of the European bison was so little, going back to 2 males to repopulate, we now can see that the cheetahs were far less, right? Just 7 cheetahs. 7 cheetahs were able to repopulate the number of cheetahs that we currently have, which are now declining. So you can see how little genetic variation they might have. Right, so as few as 7 cheetahs survived in Africa. Cheetahs eventually made a remarkable comeback. They grew to about over 100,000 by the 19th century. But stress from both human presence and faulty genes have caused their population to dwindle to fewer than 8,000 today, with decreasing numbers of cheetahs could be heading towards another genetic bottleneck. So that's genetic bottleneck. Next is gene flow. So a final factor in changing allele frequencies is a process called gene flow. So gene flow refers to situations where genes were exchanged between previously isolated populations. So genetic drift, remember, is a population, maybe a population leaving, they're isolating themselves, they no longer are able to go back, and then they are in charge of the next generation's genetic variation. Gene flow is 2 populations that were previously isolated, now have contact with each other, now are going back and forth, and their genes are flowing between the 2 populations, essentially. So if individuals in population A and population B don't normally mate with one another, then populations A and B are considered to be reproductively isolated, right? So they cannot say that there is this, giant mountain ridge and they cannot get across it, by any means, because we're imagining that airplanes and nothing like this exists, right? Hypothetically, right? And so they are reproductively isolated. Now, let's say some individuals from population A and population B, they figured out that, let's say the snow caps have melted a little bit and there is a valley and they can go right on through. Right now, these two populations have access to one another. So some individuals from population A begin mating with individuals from population B. Population B individuals are mating with individuals from population A. And now you have reproduction occurring between the two populations. So now you have gene flow. Right? This was A, this was B. They were previously isolated because there was a mountain range in the middle, and then global warming happened, and this glacier, the snow cap melted, revealing a long lost valley, and they were able to go back and forth, and now generation 2 has access to one another. Right? So that's gene flow, and my hypothetical story. Right? The last thing we're going to talk about when we're talking about forces of evolution. And again, it's math. It looks like math. Don't be scared. It's very, very easy. This is probably the only math problem-ish that you're going to have to calculate, but it's very easy. It's the Hardy-Weinberg equilibrium. So sometimes a trait is stable, or it is in a stable state of equilibrium. So Godfrey Hardy and Wilhelm Weinberg, it's German, so it's Weinberg, technically. Wilhelm Weinberg. They identified 7 key conditions that were necessary for this stability, right, in a state of equilibrium. So these 7 conditions were: 1, no mutation is occurring. 2, no natural selection is occurring. 3, no gene flow is occurring. 4, the population is infinitely large. 5, mating within the population is random. 6, all members of the population mate. 7, all members of the population produce the same number of offspring. So if all 7 of these conditions are true, then the genotype frequencies for the trait should be the same from one generation to the next. So the trait is in equilibrium. It is not evolving. So with this in mind, Hardy and Weinberg developed an equation to predict the probable genotype frequencies of a Mendelian trait in the next generation. So it's p squared plus 2pq plus q squared equals 1. And if you remember back to Algebra 1, it's like the same formula that we used, I think, in the quadratic equation. Don't quote me on that. It's been years since I've taken algebra. But it's an equation that we're familiar with, right? So p squared plus 2pq plus q squared equals 1. So the Hardy-Weinberg equation is used for simple Mendelian traits that are determined by one gene with 2 alleles. When analyzing a trait with one dominant allele and one recessive allele, P is the frequency of the dominant alleles for the trait, Q is the frequency for recessive alleles. Therefore, P plus Q equals 1, because the total number of alleles must account for the 100% of the gene pool. The chance of being homozygous dominant is P squared. The chance of being homozygous recessive is Q squared, and the chance of being heterozygous is 2pq. So the whole population must be the sum of all the possible genotypes. So p squared plus 2pq plus q squared must equal to 1, or 100% of the population. Don't worry, in the lab assignment and on your quiz, I give you the numbers. I tell you this is for, homozygous dominant, this is for homozygous recessive, this is heterozygous, calculate. And then you just got to tell me, is the population evolving? If the numbers shift at all, then the population is evolving. That's how you calculate it. It's very, very simple. The lab assignment is identical to the question on the quiz. So again, don't worry about it. The only difference is maybe your numbers might be different, but the process is exactly the same, right? Just take the numbers, plug them in, and calculate. Any change means evolution is happening. And that's it. That's it for the forces of evolution. It is a short, sweet chapter, very easy, but a lot of content in it. Again, I highly suggest going and watching the extra videos that I have posted in the notes, and I might be posting more into the module for you guys to help you all out.